Neighborhood ponds are more than just a backdrop for your summertime picnic or the vehicle for a quick canoeing session. They're bustling hubs of biodiversity. Welcome to the world of freshwater ecology. It's springtime here in Columbus, Georgia, and I, along with a team of researchers, took it upon ourselves to go on a semester-long project examining Lynn Haven Pond, which is dammed on the west side and supplied with water on the east side by a stream. We set out to determine the origin of the stream, enlisted the help of a local water specialist.
So we have our bag labeled. We also have chain of custody forms,
But our main focus was to analyze primary producers, primary and secondary consumers, and decomposition and abiotic factors of the pond. Primary producers, such as algae, were hypothesized to have slower growth in shaded areas of the pond in comparison to more open areas. It was also predicted that vegetation would differ in diversity between the north and south sides of the pond. Consumer macroinvertebrates were predicted to have the highest diversity in areas with shade, which are more complex, as well as areas with habitat heterogeneity. We predicted that the highest CPUE, or catch per unit effort, would be seen on the west deepest area of Lynn Haven Pond. Further, we hypothesized that the spring would bring about a gradual increase of temperature, and that vegetated areas such as the south or east portions of the pond would experience the highest decomposition rates and dissolved oxygen. Let's check it out.
We have about an inch, 2-inch PVC pipe, 3 of them, like so, attached with zip tie, and then we attached it to a long, really bright rope, and then we have these indicators, and hopefully we had some growth on it. So that's what we're going to be collecting our data with. And for the quadrats, we just slopped them into the water every meter, so we collected 10, counting how many species were in the quadrats. So we were able to find myrtles, pines, loblollies, elms, rushes, and then we had what we thought were green algae. It was actually, I believe it's a rhizona. It looks like snot with green speckles.
We collected our paraphytometers from the open and closed or shaded areas of the pond. Algae was scraped free from the yellow flagging tape in preparation for drying and ashing. Samples were poured through a filter, placed in a tin. First, they were dried at approximately 100 degrees Celsius, weighed afterward, then ashed at 500 degrees Celsius.
All right. Okay, so can you describe the methods that you used in your group?
Yeah, so we went to the lake and we dip netted using a D-shaped dip net. And
we took meter sticks and we pushed the sweep nets into the ground for a meter's length, and we dug down into the sediment so that we could get as much macroinvertebrates as we could out of there.
Dragonfly?
Yeah.
Dragonfly. A lot of them.
oh, one fish from a sweep,
Yeah, we got a fish.
little baby fish.
We also saw a lot of tadpoles, but we couldn't sweep them.
We pick it up and we look at the bugs inside of the water. We fed those into an ethanol little canister to save them for lab and identify them down to family.
We took the top part of the pond and the bottom part right below the dam, and we did sweep nets to get macroinvertebrates along with smaller fish. We also did minnow traps that were able to catch larger fish and even smaller fish. And then we also did electrofishing, and then we threw them back in the pond.
It's going to trap all the bugs at each of these different little spaces, and then we're going to stake them with their samples so that everything stays together. And then once we're done, you can unscrew the pieces, and we're going to take each plate and scrape off the bugs into a petri dish and see what we collected.
Okay, so for the abiotic group specifically, our methods for this project were we were going to do a
collection of things in order to collect the abiotic data, which is like anything non-living. One of our first ones was, of course, like trying to figure out the size and depth of the lake and forming kind of the profile to how deep it is in certain parts and like the concentrations of sediment and things like that. One of the other methods that we taught was rate of decomposition. That was done by putting in bags of leaf litter. They're basically just leaves that we collected from this site, put in between mesh, but left in the water for a moderate amount of time just to kind of see what the rate of decomposition is, and we'll weigh it before and after to see how much leaf litter has decomposed and how much matter we still have. One of the other methods that we also did was we deployed what are called hobos.
Hobos are small battery-powered devices which were attached to PVC pipes, constructed together, and later placed into the pond.
They measure light and temperature. We place those in varying parts of the lake, and like the deeper parts and the shallow parts, or the parts in shade and the parts not in shade, to kind of get a picture of what the temperature changes are throughout the pond, if there's any kind of like stratification or anything like that.
Start at 5 meters, guys.
And we went out into the boat and able to use a Secchi disc to measure how far light is penetrating into the lake.
So you put it in as far as you can see until you can't see the disc anymore, and that's kind of measuring like turbidity and things in the water. And that's everything for the abiotic group.
Okay.
What did these analyses tell us about Lynnhaven Pond? The paraphytometer's dry weights in the open and closed areas were not significantly different. Diversity of primary producers in the north region was much higher in comparison to the southern region. North and west both were significantly different than south and east.
With Wash Shannon's diversity, there was a significant difference, and so we ran a post hoc, and it showed that north and east are significantly different, but the south and west are not compared to north and east.
Log density per meter squared is right around 1. So I guess, I don't know, it's pretty, like the density of these macroinvertebrate families are like pretty even throughout the whole lake.
What we found was warmouth did have significance. So then we ran a post hoc test to see where it had significance, and it looks like it was in the west.
So there were more in the west.
Yeah, so we caught a lot more warmouth in the west side. CPUE, not significant.
For the two, no real difference, to be honest.
Well, and that shows over time, so that shows loss over time. So they did lose weight over time.
The percent loss, that's the percent loss. So a couple of them gained weight, don't know how. So the top one is the ones that gained weight, whereas the second box over to the far right is the ones that we removed the negative ones, because there were like 4, I think, that we had that gained weight.
Different eggs had significantly different thick disc depths.
Why would there be temporal variation in Secchi depth?
Spring rains. I mean, we were out
Rains.
there. We were rained on, yeah.
We got rained on.
The dry weights in the open area were not significantly different than the shaded area. We hypothesized that north and south would see a diversity in primary producers, which was confirmed since Shannon's diversity was significantly higher on the north side and significantly lower on the south side. We originally predicted that CPUE would be higher on the west of the pond, and sort of in support of this, the number of warmouth captured was highest in the west. The hypothesis that more species would be in the area with habitat complexity was found to be incorrect, as the north, a side with few aquatic vegetation, showed the highest Shannon diversity. And finally, we surprisingly saw that there was little difference to the decomposition rates in the shaded versus the open areas. Thank you for watching.